Hey there, I'm Rhiannon Curtis, and I'm going to be explaining DNA replication in prokaryotes. DNA replication is the process of DNA making an identical copy of itself during cell division. After considering 3 different variations of how DNA replicates itself, researchers found that it occurs in a semi-conservative manner. This process begins at the origin of replication, when an initiator protein recognizes a sequence of nucleotide bases that then signals helicase to create the replication fork, as well as elongate it as DNA replication occurs. Helicase breaks the hydrogen bonds that form the 2 strands together. As helicase moves down the double helix, strain is placed on the still bonded strands downstream of the replication fork. This strain can cause supercoiling, which is when DNA becomes overwinded. In order to relieve this tension, the enzyme gyrase moves in front of the helicase enzyme. As the 2 strands separate, this creates 2 different strands that require replication. One strand is called the leading strand. This strand undergoes continuous replication. The other strand is called the lagging strand, and it undergoes discontinuous replication. The deciding factor between these 2 strands is the direction that DNA polymerase III can move. DNA polymerase III is an enzyme responsible for most DNA synthesis. DNA polymerase III is initiated by RNA primase, which creates a short RNA sequence called a primer. This sequence is later turned into DNA. DNA polymerase III can only add nucleotides in the direction of 5' to 3'. The leading strands allows for this to happen going towards the replication fork and can thus happen in a continuous motion. Since strands of DNA match up 5' to 3', this means that the lagging strand will require DNA polymerase to move away from the replication fork. This requires the enzyme to replicate a fragment of DNA and then move backwards to replicate the next fragment, and so on. These fragments are called Okazaki fragments. These fragments are attached back together through ligase activity. Ligase is an enzyme that connects Okazaki fragments and the 2 strands by creating phosphodiester bonds. This is a bond between the 3' OH group of one nucleotide and the 5' phosphate group of another. Of course, with anything this tedious, mistakes will be made, but they are fixed through the 3' to 5' exonuclease activity of DNA polymerase III, or DNA polymerase I. It can go backwards over the newly synthesized DNA and proofread the sequence to recognize and fix any errors that occurred. DNA polymerase is an enzyme that replaces the RNA primer sequence back to DNA at the 5' end. As a new sequence of DNA is created, single-stranded binding proteins form and coat around the strand in order to protect and stabilize it, as well as prevent any of the DNA from joining together, forming a double-stranded hairpin. Termination of DNA replication occurs when 2 different replication forks meet. Lastly, if DNA polymerase I could not accurately perform its 5' to 3' exonuclease activity, the RNA primer sequence would not get changed back to DNA, and neither would the mistakes made when synthesizing the new DNA. This would cause mutations in the DNA of the organism and have effects on the replication. That's DNA replication for you.